Today our topic is about the micturition reflex. It is about the urinating and non-urinating processes in our central nervous system and in our accompanying with the bladder. So let's get started. First of all, a basic anatomy is required that the muscle lining the bladder is known as the detrusor muscle, and in the detrusor muscle there are stretch receptors, right? And they will send impulses due to the stretching and non-stretching, right? And here is the internal urethral sphincter. This is under involuntary control, and the urogenital diaphragm has two muscles. First is the external urethral sphincter, and this is under our control, it's voluntary control. And the other muscle, that is transverse kind of muscle, known as the deep transverse perineal muscle. These two make up the urogenital diaphragm. As you can see that I have drawn a empty bladder, right? The empty regards that maybe 10 to 20 ml of urine is present and it's considered to be empty, right? And the full bladder is that when the urine exceeds the capacity of 400 ml or 500 ml. The 500 ml to like 400 to 500 is the quantity when the person starts to feel to go to the bathroom, right? So let's get started. First of all, we will see that when there is little or no urine present in the bladder and there are certain impulses that will ascend and descend, and there is involvement of some kind of specialized ganglion. So let's get started. First of all, I have labeled the steps in numbers so it's easy for you to see. First thing is first that the stretch receptors are less activated, right? Because there is less amount of urine in the bladder, so stretch receptors are less activated, and the sensory afferent neurons will carry less action potential towards the spinal cord. And the second important thing is that the involvement of cerebral cortex, okay, the involvement of pons, and the involvement of thoraco-lumbar and the sacral region of the spinal cord has its great importance. The sacral region of spinal cord has about vertebra level to S1 to S4, and in some literature they say that S1 to S3. And in the thoraco-lumbar we will mostly concentrate on the last part of the thoracic vertebra, it's T11 to lumbar vertebra about L2, okay, T11 to L2, but I have drawn for convenience only one vertebra to show the sacral and the thoracolumbar region respectively. So as we're discussing that, first thing, the stretch receptors are stimulated, but it's less stimulated because the urine quantity is less, it's approximately 10. Sensory afferent neurons will carry less action potential towards the sacral region of spinal cord. Okay, here it will synapse and the fibers will ascend. In the second labeling you can see the fibers will ascend and they go to the thoracolumbar region of the spinal cord. There they will synapse with the lateral horn. And what's present in the lateral horn? They will synapse with the preganglionic motor neurons. It means that we are saying preganglionic, it means it implies that ganglion is somewhat present after, right? So these are preganglionic motor neurons present in the lateral horn of the spinal cord in the thoracolumbar region. And the third thing is that these fibers will synapse with a particular ganglion. To be more specific, it's inferior mesenteric ganglion. And this inferior mesenteric ganglion is located in the thoracolumbar region, and this ganglion is sympathetic ganglion. It's not parasympathetic. It's of great importance that it's sympathetic ganglion. The fourth thing is that the fibers from the inferior mesenteric ganglion go via the hypogastric nerve, and these fibers are postganglionic motor neurons. It means that these motor neurons will synapse with muscles, right? Because the motor fibers has functions to contract or relax some specific kind of muscles, right? The hypogastric nerve is responsible to carry the postganglionic motor neurons, and they will synapse with two particular locations present in the urinary system. First is the bladder, and to be more specific, what part of bladder, they will synapse with the detrusor muscle, and the second part is they synapse with the internal urethral sphincter. And what do they will secrete there? The sympathetic neurons, because I have said that this ganglion is a sympathetic ganglion, and these fibers are postganglionic sympathetic motor neurons. They will secrete norepinephrine always, because the sympathetic nervous system is responsible for the secretion of norepinephrine as the target organ. Norepinephrine, talking about the detrusor muscle. In the detrusor muscle, there are special kind of receptors known as beta 3 adrenergic receptors. Okay, and when norepinephrine stimulates the beta 3 adrenergic receptors, it will cause the relaxation of the bladder. In the internal urethral sphincter, there are special kind of receptors known as alpha 1 adrenergic receptors, and when norepinephrine stimulates or synapse with these receptors, it will bring about constriction or contraction. What does it mean? It means that for not to urinate, because there is small quantity of urine, we need to store the urine in the bladder, right? We do not want to pee right now, right? So the bladder will starts to relax, and the internal urethral sphincter starts to contract, because we do not want the forward flow of the urine, right? We need to store the urine right here. So beta 3 adrenergic receptors, it will cause relaxation, and alpha 1 adrenergic receptors, upon stimulation by the norepinephrine, they will cause constriction. Okay, this was the fourth step. What is the fifth step? Let's discuss. Other fibers. Other fibers will ascend, and they will synapse with the pontine and the cerebral cortex. Okay, this is the pontine, and this part of the cerebrum is the cerebral cortex. They will ascend. The sixth thing is that the fibers from the cerebral cortex, they will become aware that there is small quantity of urine present in the bladder. So now we need to store the urine. We need to store the urine, and there are specialized type of centers located in the pons. There are two types of centers. First is the pontine storage center, and the second is the pontine micturition center. What does it mean? It means that when we are planning to store the urine, we need to stimulate the pontine storage center, and we need to inhibit the pontine micturition center, because yet we do not want to micturate, right? So the sixth step is that fibers descends from the cerebral cortex, and they will synapse with the pontine storage center as well as the pontine micturition center. They will cause the stimulation of the pontine storage center, because we are storing the urine, and they will inhibit the pontine micturition center. What is the seventh step? The fibers from the pontine storage center, they will descend. They will descend towards the thoraco-lumbar region, and there they will stimulate the inferior mesenteric ganglion. What does it mean? It means that fibers from the pontine storage center, they descend, and they will synapse with the like preganglionic motor neurons, then the ganglion, and they will stimulate the action of the postganglionic motor neurons. It means that the process of the relaxation of the bladder and constriction of the internal urethral sphincter will go on. The bladder will relax, and the internal urethral sphincter will start to contract even more, because the pontine storage center has been stimulated. What is the next step? Other fibers, because the seventh step was the pontine storage center was sending impulses to inferior mesenteric ganglion. Other fibers will descend as well, and they will synapse with the lateral horn of the spinal cord. What is present in the lateral horn of the spinal cord? This is the part of the parasympathetic nervous system. Okay, these fibers are parasympathetic, nervous system fibers. The, the fibers from the pontine storage center descend and they will synapse with these fibers, the fibers of the parasympathetic nervous system, and they will inhibit it. It means that originally, when they were not inhibited, they were releasing a special type of neurotransmitter known as the acetylcholine, and the function of acetylcholine is to contract. Specifically, these receptors are present in the detrusor muscle. The receptors are muscarinic type 3 receptors, and originally acetylcholine was going there and they were causing contraction of the bladder. But now we need to store the urine. We do not want the detrusor muscle to contract, right? So this path is inhibited by the action of pontine storage fibers, right? Pontine storage center fibers, they will inhibit the parasympathetic nervous fibers. They will cause the inhibition of acetylcholine in the muscarinic type 3 receptors present in the detrusor muscles. When the acetylcholine fibers are inhibited, it means that there is no contraction but relaxation, and that's what we needed exactly. We need the relaxation of the bladder as to store the urine. This was the 8th step. The 9th step is what? The other fibers from the pontine storage center will descend and they will synapse with the anterior gray horn of the spinal cord in the sacral region. And this part of the spinal cord, or these fibers, are the part of the somatic nervous system. This is not the part of autonomic nervous system, because the autonomic nervous system comprises of two parts: the sympathetic, the inferior mesenteric ganglion, and the parasympathetic. That's what we have discussed. This part of the spinal cord, the anterior gray horn of the spinal cord, in which the fibers descending from the pontine storage center will synapse. This is the part of somatic nervous system. The fibers will go via the pudendal nerve and they will specifically synapse with the external urethral sphincter receptors. These receptors are specifically nicotine type 1 receptors, and these fibers, the pudendal nerve fibers, they release acetylcholine. And what is the function of acetylcholine? Acetylcholine is a neurotransmitter that bring about contraction, and the external urethral sphincter is contracted. It means that urine is stored because the bladder is relaxed. Internal urethral sphincter is contract and the external urethral sphincter is also contract, right? So it means that the storage function of the bladder is still going on. So let's revise this stuff. First of all, when there is little urine in the bladder, the stretch receptors are less activated and sensory afferent neurons will go toward the spinal cord in the sacral region. The fibers will ascend in the thoraco-lumbar region and they will synapse with the preganglionic motor neurons, okay, in the lateral horn. The inferior mesenteric ganglion, they have cell bodies, and from the cell bodies of the inferior mesenteric ganglion, fibers arises, which is hypogastric nerve, or we can call them postganglionic motor neurons. They will synapse with the detrusor muscle and the internal urethral sphincter. In both conditions, they will release norepinephrine. Detrusor muscle have beta 3 adrenergic receptors and they will cause the relaxation of the bladder, and the internal urethral sphincter have alpha 1 adrenergic receptors. They will bring about contraction or constriction. Other fibers will ascend and synapse with the cerebral cortex and the pontine. The fibers from the cerebral cortex will descend and they will stimulate the pontine storage center and they will inhibit the pontine micturition center. 8th step, the fibers, no, 7th step, fibers from the pontine storage center will descend and they will again stimulate the inferior mesenteric ganglion so that the relaxation of the bladder and contraction of the internal urethral sphincter still goes on. Other fibers will descend and in the sacral region of the spinal cord they will stimulate the lateral horn, they will inhibit it. On the inhibition, it will cause the relaxation of the bladder, because originally these fibers were sending acetylcholine to the muscarinic type 3 receptors located in the detrusor muscle, but now these fibers are inhibited. The ninth step is that other fibers from the pontine storage center will synapse with the anterior gray horn of the spinal cord in the sacral region. These fibers go via the pudendal nerve and they will cause the release of acetylcholine, which will act on the nicotinic type 1 receptors on the external urethral sphincter, and they cause contraction. It means that all of these processes are promoting the storage of the urine rather than micturition, right? So we have discussed that when the when there is little amount of urine present in the bladder, so how the central nervous system send impulses and receive impulses accordingly. So now let's discuss when the bladder is full. It means that the bladder is full approximately to 400 to 500, because when there is 200 ml of urine in the bladder, until appropriate time all the mechanisms are triggered that we have discussed. But when the quantity of the urine exceeds more than 400 to 500, we need to start go towards the bathroom. It means that the fibers that will promote the urinating will start to work, right? So let's discuss about it. First of all, there is a lot of stretching. There is a lot of stretching because the urine amount present in the bladder has exceeded approximately 500 ml. So urine quantity has been increased. It means that the stretch receptors will be more stimulated. Action potential will go towards the sacral region of spinal cord and the fibers will start to ascend. And these ascending fibers will bypass the thoraco-lumbar region, okay, and they directly go towards the cerebral cortex and the pons. When the fibers reach the cerebral cortex, the person starts to become aware of the accumulating urine in the bladder, okay. Second step was that the fibers ascend towards the cerebral cortex. Third step is that the fibers from the cerebral cortex will descend towards the pons, and they will promote the micturition center and they will inhibit the pontine storage center, because now we do not want to store the urine more, right? We need to micturate the urine. So micturition center has been stimulated and pontine storage center has been inhibited. Fibers from the pontine micturition center will descend. This is the fourth part, and they will go towards the thoraco-lumbar region of the spinal cord. There they will inhibit the inferior mesenteric ganglion. They will inhibit the preganglionic motor neurons. It means that the ganglion itself is inhibited, and the postganglionic motor neurons, that was hypogastric nerve, right? It is also inhibited. This part and this part. It means that originally they were releasing norepinephrine to the detrusor muscle and to the internal urethral sphincter via the beta 3 adrenergic and alpha 1 adrenergic respectively. But now, as we have said, that the fibers from the pontine micturition center has inhibit the preganglionic motor neurons. So this all process is inhibited. It means that the bladder relaxation is inhibited and the internal urethral sphincter contraction has been inhibited. So what is the process? Now the bladder starts to contract and the internal urethral sphincter starts to relax, and that's exactly what we need in accordance with the micturition, right? The sixth step is that the fibers will descend more. Other fibers from the pontine micturition center will descend, okay, the orange line, and they will synapse with the lateral horn. They will synapse with the lateral horn. These were the parasympathetic motor neurons, okay, these are the blue one are the parasympathetic motor neurons. They will go towards the detrusor muscle. Originally, they were releasing acetylcholine, but originally we have inhibited that, right? In the case of storing the urine, we have inhibited this acetylcholine muscarinic type 3 receptors. We have inhibited that in accordance for the relaxation of the bladder. But when we start to urinate and when we are planning to urinate, we need to contract the bladder, right? So this process is promoted or this process is stimulated. It means that muscarinic type 3 receptors that were located in the detrusor muscle, they were stimulated by the action of acetylcholine. And what is the function of acetylcholine? The function of acetylcholine is to contract the muscles. So bladder starts to contract, it means detrusor muscle starts to contract. Last but not the least, it was the pudendal nerve, right? The seventh is the pudendal nerve. Fibers from the anterior gray horn. These fibers were originally descending from the pontine and the cerebral cortex, right? Fibers from the cerebral cortex, they synapse with the anterior gray horn of the spinal cord, and the motor fibers will go via the pudendal nerve. These fibers were originally stimulated because the external urethral sphincter starts to contract, and that's what we needed in the storing part. But in urinating part, we need to relax the external urethral sphincter, so we need to inhibit the acetylcholine. And what was the receptors here? These were nicotinic type 1 receptors, right? So this is inhibited, and this is inhibited, it means that relaxation will start. And when relaxation starts, urine will start to flow in the forward direction. Okay, let's revise this a little bit. First of all, when the urine quantity starts to exceed, the stretch receptor starts to have more stretch. Okay, when there is more stretch on the stretch receptors, they will cause increased action potential. They go towards the sacral region. Fibers will ascend. They will synapse with pontine and the cerebral cortex. Cerebral cortex become aware that of the accumulating urine in the bladder. Fibers will descend and they will promote or stimulate the pontine micturition center and inhibit the pontine storage center. Fibers from the pontine micturition center will descend and they will synapse with the preganglionic motor neurons in the thoraco-lumbar region and they will inhibit it. It means that all the activities that were responsible from the inferior mesenteric ganglion has been inhibited. So bladder relaxation and the internal urethral sphincter bladder contraction and the internal urethral sphincter relaxation starts. Other fibers will descend and they will synapse with the lateral preganglionic motor neurons. It will cause contraction. Muscarinic type 3 receptors are present in the detrusor muscle. Acetylcholine will be released and it will cause the contraction of the bladder. The last part is that the pudendal nerve fibers, they will synapse with the anterior gray horn of the spinal cord. Pudendal nerve will cause inhibition of the nicotinic type 1 receptor. So acetylcholine will have no effect. It means that relaxation starts. And that's what we need. Contraction of the bladder, relaxation of both sphincters, internal as well as the external. So the person starts to urinate, and when the person has urinated, again the bladder starts to store the urine until 500 ml is reached. So this is all the process that is repeated all the time when storing and urinating processes of the urine. This was for today. I hope you understand the stuff relating the micturition reflex, and I will see you in the next video. Till then, goodbye.